This is the second unit, or the second lecture in Unit 1 for microbiology, the chemistry review. There are several animations that we will be viewing in this presentation that you'll be able to find the links in this document in Angel, and you'll also be able to find them under the list of website links so that you can go in and watch them on your own later on. Anything that takes up space and has mass is considered matter. The smallest chemical unit of matter is the atom. For any references to chemistry made in this class, the Bohr model of the atomic structure from 1913 will be adequate. The protons, neutrons, and electrons are the subatomic particles which make up the atom. Protons are positively charged and are found in the nucleus. The neutrons are uncharged or neutral and are also found in the nucleus. The electrons have a negative charge and are found in the shells surrounding the nucleus. The most significant of these shells is the outer valence shell. Elements are composed of a single type of atom. The elements are represented by the different chemical symbols on the periodic table. The atomic number tells us the number of protons in the nuclei. If the atom is neutral in charge, there will be an equal number of electrons to the protons, where ions will have a different number of electrons than protons, giving them a charge. The atomic mass is the sum of the masses of all the subatomic particles. Of the particles, the electrons are the smallest. An isotope occurs when an atom of a given element occurs with a different number of neutrons in the nuclei. This is different from the ion, which has a different number of electrons. The electron configuration or arrangement of the electrons determines the chemical behavior. The valence electrons are those in the outermost shell. These will be the ones that interact with other atoms and determine its combining capacity. Here is a video that you can watch on isotopes on YouTube. You should be able to follow the link. It is also under the website links in Angel for this section. There are two main branches of studying chemistry, inorganic and organic chemistry. Organic chemistry is the study of carbon and hydrogen. Inorganic chemistry studies all of the elements, including carbon and hydrogen, but they are not central to inorganic chemistry. The terms molecules and compounds are commonly used interchangeably, but do have distinct meanings in chemistry. A molecule involves two or more atoms of the same element, while a compound is two or more atoms with different elements. We commonly say water molecules, but water is a hydrogen and oxygen, making it a compound. Although this distinction is mentioned, it is not adhered to in this or many other biology-related classes. Atoms will interact with neighboring atoms or form chemical bonds. There are a few variations of these chemical bonds we will look at. In the covalent bonds, electrons are shared by two atoms. Remember, in the Bohr model, the first shell can accommodate 2 electrons, and the second and third shells can each accommodate 8 electrons. When the atoms share electrons, they can share them equally, or one can dominate most of the time with the electron in its field. In a polar covalent bond, the atoms have different electronegativities, or pull on the electrons, so the electrons will spend more time around one of the atoms. This can create partial charges on different sides of the atoms. In a nonpolar covalent bond, the electronegativities are similar, and the electrons will spend equal time around both atoms. With ionic bonds, there is an attraction between positive and negative charged ions. The ions are created by either gaining or losing electrons in the valence shell of the atom. The loss of electrons is seen most often when there is only 1 or 2 electrons in the valence shell, and the gain of electrons is seen most often when there are 7 or 8 electrons in the valence shell. The atom will gain or lose electrons to reach a more stable state with 8 valence electrons. The cation is the positive charged ion, while the anion is the negative charged ion. Together they will form salts or a crystalline form of the compound in the solid state. When they are dissociated in water, they are known as electrolytes. Hydrogen bonds are the result of attraction between the partial positive and partial negative charges. Here we will watch a brief tutorial on chemical bonding. And so it appears the hot link is not going to work for me. And this is probably why. So here we have an introduction to the chemical bonds. If we take a purely mechanistic view of life, we can consider each living cell as an intricate web of chemical reactions. Atoms combine with other atoms to form molecules and then recombine again during other chemical reactions to form different molecules. These chemical reactions are the basis of a cell's structure and metabolism. In the accompanying animation, we examine chemical bonds, which are the attractive forces between atoms. We focus in particular on the role of electrons in chemical bonds. The behavior of electrons explains most aspects of how atoms interact with each other in the living and non-living world. Here we'll click on the Animation tab to study the properties of covalent, ionic, and hydrogen bonds.
The chemical reactivity of an atom is based on the number of electrons the atom contains in its outermost electron shell. Electron shells are depicted here as circles around the nuclei of the atoms. Each shell contains electrons with particular energy levels. Let's examine the electron shells of the element argon. The first electron shell holds just 2 electrons. These are electrons at the lowest energy level, the shell that electrons of an atom fill first. The next 2 shells can each hold 8 electrons. Argon's third and outermost shell is already filled to capacity, making this element extremely unreactive or inert. Chlorine also has electrons in 3 electron shells, but its third shell contains only 7 electrons. In chemical reactions, chlorine has a tendency to gain an electron, filling this shell with 8 electrons. This tendency to fill the outermost electron shell with 8 electrons is known as the Octet Rule. Sodium has electrons in 3 electron shells, but its outermost shell contains just 1 electron. Sodium cannot receive 7 additional electrons to complete this shell. This would require a huge amount of energy. Instead, sodium has a tendency to lose its outermost electron, leaving this atom with 2 filled inner shells. In general, elements with filled outer shells are inert, but atoms of all the other elements are chemically reactive and can form chemical bonds with other atoms. A chemical bond is an attractive force between atoms. Bond formation relies on the ability of atoms to share, donate, or receive electrons from other atoms. Let's examine the nature of a chemical bond, using hydrogen atoms as examples. 2 atoms are depicted with their electron shells. 2 others are depicted more realistically, showing a region of space around the nucleus where the electron has a high probability of being found. This region is an electron's orbital. If 2 hydrogen atoms approach each other, the positively charged nucleus of one is attracted to the negatively charged electron of the other. The orbitals merge, and the electrons are then equally attracted to and shared by the 2 nuclei. The electron-rich region between the nuclei binds the atoms together. A covalent bond results when two atoms share electrons. In the case of two hydrogen atoms, each shares its single electron with the other. This sharing allows each to fill its electron shell with two electrons. The pair of shared electrons constitutes a covalent single bond. Let's now consider oxygen, an atom with 8 electrons. 2 electrons fill the innermost shell, and the other 6 electrons reside in the next shell. This outer shell needs 2 more electrons to complete it, the Octet Rule. 2 oxygen atoms form a covalent double bond by sharing 2 electron pairs from their outer shells. Carbon is perhaps the most versatile element on Earth, in large part because it contains only 4 electrons in a shell that can hold 8. To fill its outer shell, carbon forms 4 covalent bonds with up to 4 other atoms. In a molecule of methane, carbon shares electrons with hydrogen atoms, forming 4 covalent single bonds. Although this molecule is relatively simple, carbon often forms the backbone of large, complex molecules. With each carbon atom able to bond to 4 other atoms, carbon-based molecules are incredibly diverse. Triple bonds are rare, but nitrogen gas molecules, the most abundant molecule in the air we breathe, form triple bonds. The 2 nitrogen atoms share 3 pairs of electrons, allowing each to have 8 electrons in its outermost electron shell. In a covalent bond, 2 atoms may share electrons equally or unequally. In cases in which both atoms are of the same element, such as in molecular hydrogen or molecular oxygen, the electrons are shared equally by each atom. Sharing may be unequal between atoms of different elements, such as in the carbon and hydrogen atoms of ethane, or the hydrogen and oxygen atoms of ethanol. For these molecules, we need to consider a concept called electronegativity, the ability of an atom to attract electrons. Oxygen has a high electronegativity, meaning that it strongly attracts electrons. Hydrogen has a much lower electronegativity. The shared electrons are drawn closer to the oxygen atom of ethanol. Because oxygen draws the negatively charged electrons closer to itself, it becomes slightly negative in charge, indicated by delta minus. Hydrogen, which doesn't hold the electrons as closely, becomes slightly positive in charge, indicated by delta plus. A covalent bond in which electrons are not equally shared is a polar covalent bond. Now let's examine the covalent bonds in ethane. All of the bonds are formed between carbon and hydrogen, which have similar low electronegativities and attract shared electrons nearly equally. No atom in ethane takes on a significant partial charge. The bonds between the atoms are nonpolar covalent bonds. The polar nature of the bond between oxygen and hydrogen in ethanol results in a polar molecule that can interact with other polar molecules. The hydrogen with a partial positive charge is attracted to an atom with a partial negative charge on another polar molecule, such as water. This kind of attraction is a hydrogen bond. The structures of many molecules, as well as their ability to dissolve in water, are based on the ability to form hydrogen bonds. A DNA molecule, for example, contains 2 strands of atoms. The bonds within each strand are covalent, but the 2 strands associate with each other through hydrogen bonds. The ability of an atom to attract electrons, its electronegativity, might be so strong that the atom does not share electrons with other atoms, but instead strips electrons away from other atoms. Chlorine has a high electronegativity, whereas sodium has a very low electronegativity. Because sodium has a low electronegativity, it holds its outer electron very loosely and easily loses it to chlorine. When this happens, chlorine fills its outer shell with 8 electrons, becoming negatively charged. Sodium loses its electron and becomes positively charged. Atoms that have lost or acquired electrons are called ions. Positive ions, cations, and negative ions, anions, form ionic bonds by an electrical attraction. In solid form, the ionic bonds between sodium and chloride ions are very strong because the ions are so close together. These ions make up table salt. Ionic bonds can be broken down in water. The partial charges on water molecules are attracted to the full ionic charges on the ions. When the solid dissolves, it breaks down into separate ions, each surrounded by a sphere of water molecules.
Two main properties influence how or whether bonds form between atoms. One, the state of an atom's outermost electron shell determines whether an atom is reactive. Helium and neon have outer shells that are completely filled with electrons, and for this reason these atoms are inert. If an atom has partially filled the outer shell, however, it is its tendency to form bonds with other atoms. The second main property is the atom's electronegativity, which influences the kind of bond an atom forms. If two atoms have similar electronegativities, they tend to share electrons equally in nonpolar covalent bonds. However, if electronegativities differ, then the atoms tend to form either polar covalent bonds or ionic bonds. Bonds between atoms in molecules also influence how these molecules interact with other molecules. For example, if a molecule contains polar covalent bonds, then the molecule itself is likely polar. A polar molecule can form hydrogen bonds with water molecules and therefore dissolve in watery milieu of a cell. Once dissolved, it can easily find and interact with other molecules and thereby participate in the cell's metabolism. Hydrogen bonds are also important because they hold together regions of large molecules such as DNA or proteins, maintaining the shapes and functions of these important biomolecules. There is a little quiz section here that you can take on your own. Remember, this link should also be available in Angel as well as in the Tegrity presentation itself. Chemical reactions begin with the reactants and end with the products. The reactants are the atoms, ions, or molecules at the beginning on the left side of the arrow, and the products are the atoms, ions, or molecules at the end on the right side of the arrow. There are several categories of chemical reactions, and we will look at a few examples. In a synthesis reaction, larger, more complex products are formed. In a dehydration synthesis, two small molecules are covalently joined as a result and produce water. A reaction which requires the input of energy is endothermic. Synthesis reactions in organisms are known as anabolism. Decomposition reactions break larger molecules or compounds into smaller ones. They are generally exothermic and release heat. Hydrolysis is an example of decomposition, in which a covalent bond is broken and the ionic components of water, the hydroxide and hydrogen ion, are added to the products. Decomposition reactions in organisms are known as catabolism. In an exchange reaction, we see the transfer of atoms where they are formed from one molecule to another. An example would be substance A reacted with substance BC, forms substance AB plus substance C. Water makes up 50 to 99% of most organisms. It has two polar covalent bonds which give it several unique properties necessary for living organisms. These bonds give it cohesiveness to hold water together and create surface tension. It is a good solvent. Like dissolves like, so many ionic substances can dissolve in water. It absorbs heat slowly and has a wide temperature range in the liquid state. It will participate in many chemical reactions as well as being a medium for reactions to occur in. Acids and bases are created when water is split into its ionic components. Acids dissociate into one or more of the hydrogen ions and one or more anions, such as hydrochloric acid or sulfuric acid. Bases are molecules which will bind with the hydrogen ion when dissolved in water, such as the hydroxyl ion. The pH scale is a logarithmic scale indicating the potential or percent hydrogen. For each one whole number increase, there is a tenfold increase in the acidic or basic nature. These may involve salts, which are compounds which dissociate in water. Because the pH helps to regulate many biochemical reactions in living organisms, it is important to have a good regulation of the pH. Buffers are substances that prevent drastic changes by helping to absorb pH changes. Here on the right is a pH scale running from 0 to 14, along with some different examples of different substances in the different pH ranges. We're going to watch a little animation here on buffers.
A buffer is an aqueous solution containing both a weak acid and a weak base that are a conjugate acid-base pair. The buffer shown contains the weak acid acetic acid and its conjugate base acetate. These species are dissolved in water. Water molecules are not shown in this animation to draw focus to the buffer components. A buffer solution has the ability to resist changes in pH upon the addition of small amounts of either acid or base. Choose to add a strong acid or a strong base to the buffer by clicking one of the buttons. Only hydrogen ions, H+, from the hydrochloric acid are shown entering the solution. Chloride is not shown because it is a spectator ion. It does not participate in the reaction. The strong acid H+ reacts with the base component of the buffer acetate, CH3COO-, reducing the acid component of the buffer acetic acid, CH3COOH. The buffer has resisted a change in pH by removing strong acid H+ from the solution. Only hydroxide ions, OH-, from the strong base sodium hydroxide are shown entering the solution. Sodium ions are not shown because they are spectator ions. They do not participate in the reaction. The strong base hydroxide, OH-, reacts with the acid component of the buffer acetic acid, CH3COOH, reducing the base component of the buffer acetate, CH3COO-, and water. This buffer has resisted a change in pH by removing the strong base, hydroxide, from the solution.
The organic macromolecules are larger molecules we will see in living organisms. In organic chemistry, we study the functional groups which commonly appear together. Some examples of these are methyl groups, which are CH3, or acid groups, which are COOH. Monomers are the simpler subunits, such as protein, carbohydrate, and nucleic acids. The monomers can join together to form chains of polymers. Lipids are non-monomers and are almost entirely carbon and hydrogen, making them hydrophobic. They are hydrophobic because of the nonpolar covalent nature of their bonds. Fats are made from dehydration synthesis and commonly form triglycerides. A triglyceride is a glycerol backbone with 3 fatty acids attached. In fats, we are concerned about the saturation or amount of hydrogen in the carbons. When they are saturated, each carbon is attached to the maximum number of hydrogens and only has single bonds. When it is monounsaturated, there is one unsaturated bond, which will be a double or triple bond. When it is polyunsaturated, there are multiple unsaturated bonds. Typically, the more saturated the fat, the more stable and the less likely to interact with its environment. Here shows a comparison of a saturated and unsaturated bond. So you can see where it's got a point of unsaturation here, there is a bend. And in here we can see the hydrophilic end and the hydrophobic end of a a phospholipid. Phospholipids consist of two fatty acids attached to a glycerol with a phosphate head on one end. The phosphate end is hydrophilic, while the fatty acid tails are hydrophobic. These are used in cell membranes as a phospholipid bilayer. Waxes are made from one long-chain fatty acid covalently linked to an alcohol by an ester bond. Waxes are completely water-insoluble. Certain organisms, such as the mycobacterium, make waxy walls and need special stains as a result. The sterols are made of 4 rings each with 5 to 6 carbons attached to each other. Cholesterol is the most common sterol, although there are others in living organisms. The carbohydrates are composed of carbon, hydrogen, and oxygen in a repeating pattern of CH2 n times. They are used for energy, making the backbone of both RNA and DNA, and a component of cell walls. A monosaccharide is typically a simple sugar and involves just one sugar. Their names usually end in OSE or OS and are commonly hexoses with 6 carbons or pentoses with 5 carbons. Disaccharides are made from 2 monosaccharides linked together, and polysaccharides are several monosaccharides linked together. Polysaccharides may be branched or unbranched, and may be linked with amino acids in bacteria. Peptidoglycan is an important carbohydrate in microbes. Proteins are made from carbon, hydrogen, oxygen, nitrogen, and sulfur. They are used to make structures and act as enzymatic catalysts, perform regulatory reactions, hormones, genes, transport proteins, antibodies, complement, bacteriocins, plus many other things. The amino acid is a protein monomer in this case. Their unique shape allows them to form stereoisomers. This is similar to being right or left-handed. Stereoisomers exist in the D or L forms. Amino acids are almost always in the L form. Amino acids are held together by the peptide bond, which uses a dehydration synthesis to form covalent bonds. So here you can see you've got amino acid 1 and amino acid 2. The dehydration synthesis is pulling off a hydroxyl with another hydrogen on the other side to form H2O or water. Here we're going to watch a little animation or tutorial on chiral molecules. So here you can take your own time and go through this little game on whether or not a molecule is chiral. Proteins are formed by the joining of unbranched polypeptide chains linked in a specific order. If the amino acids are in a specific order, they will always spontaneously fold up to form the same shape. The structure of a protein is broken down into 4 major components. The primary structure of an amino acid is the sequence of the amino acids. As long as the sequence is the same, the protein made will always form to the same shape. The secondary structure is the result of the ionic and hydrogen bond, and the same sequence will always spontaneously fold into the same shape. Hence, they will fold into repeating shapes of alpha helices or beta-pleated sheets. The tertiary structure gives a 3D shape, but is not repetitive in nature like the secondary structure. Here, the R groups in the amino acids will form covalent and ionic bonds like disulfide bridges. The quaternary structure will involve 2 or more polypeptide chains coming together. Here you can see the initial sequence is your primary structure. The beta-pleated sheets or alpha helices are the secondary, then folded into the tertiary, and finally multiple of them coming together for the quaternary structure. And here's another animation on the peptide bond. So again, you will have access to viewing that animation on your own as well. The DNA and RNA are made up of nucleic acids. The nucleotides are a monomer that consists of a phosphate, a pentose sugar, and a nitrogenous base, as in adenine, thymine, cytosine, guanine, or uracil. The purines are the bases with double rings. A and G are adenine and guanine, and the pyrimidines have single rings, cytosine, thymine, or uracil, or CTU. They always pair C and G together and A and T together in DNA. In RNA, A will pair with U instead. Double-stranded DNA will always run in anti-parallel strands. The 2 strands will be running in opposite directions from the 5' end to the 3' end. DNA always runs 5' to 3'. The 5' end has a carbon number 5 attached to a phosphate or has a terminal phosphate on carbon number 5. The 3' end is attached to a hydroxyl group or has a terminal hydroxyl on carbon number 3. And so there was another animation on the protein structure. When a protein is disrupted or loses its shape, we refer to it as denaturation. It can be permanent change or temporary. Things that may cause this are heat, pH changes, concentration of salt, or anything that interrupts the bonds holding the shape. The same high-energy compound, ATP, you learned about in the body is found in microbial cells as well. It is composed of adenine, ribose, and 3 phosphates. It is used as the energy currency by the cell. This is the end of the chemistry review.